Good afternoon. I am Dr. Kara Wilder, a technical writer at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Introduction to ATCC Microbiome Standards, an end-to-end solution for the standardization of microbiome research, presented by Dr. Dave Matar and Mr. Nick Greenfield. Dr. Matar is a lead scientist and the head of the microbiology R&D group at ATCC. At ATCC, he has been instrumental in the development of controls and standards for various infectious microorganisms with applications in the area of molecular diagnostics and clinical, food, and pharmaceutical microbiology. He led the development of the ATCC microbiome standards with the goal to help microbiome researchers standardize their methods. Mr. Greenfield is the founder and CEO of OneCodex, which is a microbial genomics software company based in San Francisco, California. OneCodex is the leading microbial genomics and metagenomics platform and has received awards from the Centers for Disease Control and Department of Defense, as well as been recognized by the White House National Microbiome Initiative. At OneCodex, Nick leads the development of the company's bioinformatics and software technology and oversees the company's efforts in application areas including infectious disease research, microbiome R&D, and public health. In the following webinar, they will discuss the need for standards in microbiome research, the development and use of the ATCC microbiome standards, and how to analyze these standards using the OneCodex data analysis platform. If you have any questions for either speaker, please use the questions box available through the webinar program. Questions will be answered via email and will be archived with a recorded webinar presentation on the ATCC website at www.atcc.org.
Thank you, Kara. Hello, everybody, and thank you for joining us for this webinar on ATCC microbiome standards. My name is Dave Matar, and I'll be providing you an overview about microbiome standards in this webinar. Before we jump into the microbiome world, I'll give a brief introduction about ATCC. ATCC was founded in 1925. ATCC is a non-profit organization with headquarters in Manassas, Virginia, and another R&D center in Gaithersburg, Maryland. ATCC is world's premier biological material resource and standards development organization. We have a collection of about 5,000 cell lines, around 80,000 microorganisms, plus we offer genomic and synthetic nucleic acids, and of course media and reagents to support growth of the bacteria and cell lines. ATCC collaborates and supports the scientific community and works closely with standards organizations to develop innovative biological solutions. We have an ever-growing portfolio of products and services. We have sales and distribution network in 150 countries, and we have about 14 international distributors. We are a talented team of more than 450 employees, and about 1/3 of our employees are with advanced degrees, PhDs. So with this background about ATCC, here is the outline of today's webinar. I've divided it into 4 sections. In the first section, I'll introduce microbiome research and what is the need for standards. In the second section, I'll discuss how we develop those standards. And in the third section, I'll show you some proof of concept experiments which really show how you can evaluate the biases and how you can use ATCC microbiome standards to standardize your methods. And in the last section, Nick Greenfield from OneCodex will talk about data analysis platform. Microbiome has been a very popular word lately, and I think everybody is aware that it is essentially the collective genomes of the microorganisms that live inside and on the human body, or in general in any environment. To give you some interesting facts, the total wet weight of microbiome in our body can range from 1/2 to 2 kilograms. The total number of bacteria in our gut is about 10 times greater than the total number of somatic and germ cells in our body. The number of functional genes in gut microorganisms is about 150 times higher than the total number of human genes. And Each individual has a unique signature gut microbiome as personal as a fingerprint. So microbiome overall has been a hottest area of research as of now, and it's been related to every aspect of human health and disease. As of last week, there were almost 35,000 publications on PubMed, and of that, about 80% have been in the past 5 years. In 2016 alone, there were 24 new startup companies focused on microbiome diagnostics and therapeutics. Clearly, it is one of the promising areas of research with a very diverse set of applications. The basic research on microbiome associated with the human body, such as gut, skin, oral, and respiratory, has led to a lot of insight into functional role of these microorganisms. The research is now geared more towards translational microbiome, and the role of microbiome has been implicated into various diseases such as diabetes, diarrhea, Crohn's disease, obesity, neurological disorders such as schizophrenia. There is a plethora of ongoing research, and every day I see new publications showing association of microbiome with anything and everything related to human body and behavior. I'm sure we'll see a study soon showing that people who post on Facebook more frequently and tweet more frequently have different microbiomes than the people who do not use social media. Now, what are the challenges? Despite plethora of research, publications, and applications of microbiome, the methods in the microbiome are not standardized. The currently used methods such as 16S ribosomal RNA amplification followed by sequencing or shotgun metagenomic sequencing methods, they are not perfect. There have been numerous studies showing the challenges with the current technology and the methods and the need for standardization, and I have listed some of the screenshots here from those studies. In addition, various key opinion leaders and organizations such as NIST, ABRF, Metagenomic Research Group, they all are pushing for standardization of microbiome methods. In a typical microbiome study, the main steps in the workflow include sample collection, DNA extraction, library preparation, sequencing, and data analysis. Now, all these steps can lead to bias, and there is a standardization needed for each step. Let's talk about each step. First step is sample collection. Storage of sample handling and processing can impact the outcome of microbiome. Second one is most important and often neglected step, which is DNA extraction, which can tremendously impact the outcome of microbiome and can affect the relative abundance because of incomplete cell lysis and the DNA recovery. Thirdly, various library preparation methods such as primer-based methods or amplification-based methods versus non-amplicon-based methods, different library preparation protocols and kits, they can impact the outcome of your microbiome experiments. Further, the sequencing platform, the choice of platform, depth of coverage, chemistry, all these variables can impact the microbiome readout. And lastly, not the least, is the data analysis. Choice of data analysis, what algorithm you are using, read quality, and what database are you using, it can definitely impact the outcome of any microbiome study. This bias introduced in each step was highlighted in various publications that I showed you in my last slide. One of the major challenges in standardization is the lack of controls, lack of reference materials which are required for monitoring full process as well as for developing new assays for microbiome technology. Now, for a while we did not have fully characterized process controls that can monitor the full process. We did not have good reference materials where you can use to standardize the methods or steps in each protocol. In the next few slides, I'm going to show you some of the biases we observed when we started working on microbiome. Last year, when I started working on microbiome, first thing that we did in our lab was we took about 20 bacteria from ATCC collection and we mixed them. And we extracted DNA from those mixed 20 bacteria using two popularly used kits, Kit A and Kit B. That's what I'm calling them in this webinar. And we ran shotgun analysis after extraction and purification of DNA from those mixture of 20 bacteria. On the left side is what we were expecting in the bar graph, whereas on the right side, kit A and kit B show what we observed. The relative abundance and the detection of all 20 bacteria was all over the place. We could not even detect all 20 bacteria in this first experiment that we did after doing basic extraction and using shotgun metagenomic sequencing from those extracted DNA. The next one is we extracted genomic DNA from those 20 bacteria, and we now mix the genomic DNA to make a genomic DNA mark community, just to make sure if we use any standardized protocol, can we get the same results. What we did was we sent this genomic DNA-based mark community to 3 independent labs. We told them use a standard protocol. We gave them Earth microbiome primer sets which target V4 region of 60ness, and we told them to run standard Earth microbiome protocol, analyze the data, and send the data back to us. And lab 1, lab 2, and lab 3 bar graphs show what we received from them. It was very interesting because none of the lab could detect all 20 bacteria. Everybody missed at least 1 bacteria, and one of the labs missed 4 bacteria out of 20, which clearly shows that the library preparation and the sequencing methods, they were different and which could give different readouts for microbiome analysis. Then further, we use another mark community, same 20 genomic DNA, but in this case, we use 3 different variable regions for 60ness. We targeted V1, V2, V3, V4, and V4 regions independently using the same mark communities, and we saw huge variation in the results. Out of 3 primer sets tested, only 1 primer set V1, V2 could detect 20 bacteria. Other 2 primer sets could not even detect the 20 bacteria which we were expecting in the mix. The last thing which we did to really show the bias and to figure out the bias was we used a standardized data sets from one of the mixtures that we made. We used a shotgun data set from a mixture of 10 genomic mark communities, and we simply took the same data set FASTQ files and analyzed in 2 different platforms, platform A and platform B, and we saw entirely different results. With one platform, we could not even detect 10 organisms that was present in the mix. So all these experiments that I showed you, they all examples prove that there is a lot of discrepancies and shortcomings in the current methodology, and there is a need for standardization, and there is a need for developing a reference material which can be used for standardization. So this really prompted us to develop something to meet the needs for assay optimization, or which can be used as daily run controls. Now this slide shows what we developed. We developed 2 kind of mark communities, one for helping to develop assay and standardize various steps in the assay development. These mark communities were in the form of mixed genomic DNA, and the second one was mixed bacterial whole cells. These mark communities could be used as full process control, starting from DNA extraction to all the way to analysis, which I'm showing in this slide. This slide sort of gives you an overview of 3 main steps in a microbiome workflow, which are DNA extraction, sequencing, and the analysis. And we have developed controls to optimize each step in the microbiome workflow. For example, for whole cell standards, we've developed 2 mark communities, MSA 2002 and 2003. You can use these whole cell standards to monitor the full process, starting from extraction, sequencing, to all the way to analysis. The second one are 4 genomic DNA standards, where we mixed 10 and 20 genomic DNAs in equal and staggered proportion. These 4 genomic mixtures can be used to evaluate bias in various steps during sequencing, library preparation, and all other steps post DNA extraction. And finally, we have developed not only the physical standards, but we've also worked with one codex on data analysis, because no matter how good your standards are, if your data analysis is not standardized, you are not going to get perfect results. So Nick Greenfield is going to talk about this data analysis which we have developed in collaboration with one codex. With that background of bias and the need for standardization, now I'm going to switch gears and show you how we develop these standards. The first slide shows the contents or the bacterial strains we used for making these mark communities. We basically used one of the mark communities from Human Microbiome Project, which were earlier distributed under BEI resources under ATCC. We used these HMP mark communities as template, and we further refined them and updated the strains by using ATCC fully sequenced and characterized strains, for which the reference genomes and 16S sequence are fully published in various databases. These diverse strains were selected based on their gram staining properties, GC content, genome size, number of 16S copies, and other special features that makes the mixture very diverse. The diversity leads to challenges in the DNA extraction and the sequencing methods and the analysis method. So we deliberately use very diverse set of bacteria so that we can challenge the DNA extraction and sequence method and further analytical methods to figure out the standardization of microbiome. In the next section, I'm going to show you some of the challenges which we faced when we were developing these standards. First, I'm going to talk about whole cell standards. As I mentioned, we picked up ATCC authentic cultures which were fully characterized and sequenced, and we used these standardized strains. The first thing where we got challenged was, what is the precise method for quantification? We compared different methods of quantification. CFU is one of the gold standard in quantification of bacteria. Now, CFU, although it is a gold standard, however, CFU method cannot discriminate between live and dead bacteria. In this case, since our readout depends upon the DNA, and DNA could be contributed by compromised or dead cells as well, so for that reason, we looked at other methods such as flow and image cytometry, which can detect total number of cells, especially after you stain with a DNA dye or any whole cell dye. So we did a lot of validation in comparing different methods, and especially with imaging and flow cytometry. Despite the accuracy of flow and image cytometry, we did face a lot of challenges because of diverse characteristics, shapes, clumping behavior of different bacteria in the mix. So we did end up using image cytometry after extensive optimization of each bacteria with respect to image analysis. So we invested a lot of effort into automated counting with image cytometry, and we developed a lot of software algorithms for counting each individual bacteria based on shape, clumpiness, and other morphological characteristics. Finally, we debated on what would be the best preservation method for providing the whole cell product. We tested various methods and compared DNA extractability from individual bacteria and from mixed mark communities. We could not find any significant difference between quantity and quality when we compared live, freshly grown bacteria, frozen bacteria, and lyophilized bacteria. We also compared inactivated bacteria with live bacteria. However, we found that with inactivation, there was a lot of variations in the quantity and quality of bacteria. So we ended up using live bacteria in lyophilized format. The reason we picked up lyophilized formats in addition to quality and quantity was lyophilized product can be stored at 4 degrees and can be shipped at room temperature. And finally, we made the precisely counted bacterial cell as a mark community mixed in equal proportion for whole cell mark communities. The next one is development of genomic DNA standards. We use the same strains that we use for whole cells here, again, fully authenticated and characterized ATCC strains. And we used our proprietary DNA isolation methods to extract high-quality DNA from these strains. The next challenge was, what is the best quantification method for DNA? We really compared various fluorescent dye-based quantification methods such as picogreen and qubit against other methods such as digital PCR as well to calculate theoretical and observed genome copy numbers. We found a good correlation between the quantification methods such as qubit, picogreen, and digital PCR. And the first set of 4 genomic standards we developed, we used fluorescent dye-based quantification method to calculate genome copy number for each DNA, and we mixed them in equal or staggered proportions based on the genome copy number. So with that background about how we developed this standards, in the next section, I'm going to show you some of the used cases or proof of concept experiments to show how can you use those whole cells and genomic DNA mark communities to standardize various steps in the microbiome workflow. The first and foremost is the DNA extraction step. What we did was we used whole cell mark communities, which was a mixture of 20 bacteria, and we tested 3 popularly used DNA extraction kits which are published in microbiome literature. As I mentioned, each whole cell mark community contains 2 million cells of each bacteria, and you can directly use these lyophilized cells by reconstituting and by aliquoting. The left side shows the DNA extraction data in terms of quantity and quality from 3 different kits. As you can very well see, there was quite a bit of variation in the quantity and quality of DNA which we recovered using 3 different DNA extraction kits. Further, we used this extracted DNA to perform shotgun metagenomic analysis. And you can very well see in this bar graph how much is the variation among the 3 DNA extraction kits we tested. The same data in the form of % number of reads is presented in this table. I've highlighted the bacteria which shows significant differences. For example, Helicobacter was barely detectable with kit B and C, whereas number of reads for Porphyromonas were close to expected with kit A and B. However, kit C showed more than 1 log difference. Some of the bacteria were definitely overrepresented, such as Bacteroides, Streptococcus mutans, and Deinococcus. Next, we used the same set of whole cell standard, which was a mixed mark community of 20 bacteria. And here we tested them with both 16S methodology as well as shotgun methodology. And here we used, again, 2 very popular DNA extraction kits. And the data is presented in the bar graph. Left bar shows what we were expecting, and the other bars show what we observed with 2 kits, kit A and kit B, with 16S and shotgun methods. In this experiment, we observed, this is the data from number of reads, % number of reads. And in this case, we found that gram-positive bacteria such as Bacillus, Lactobacillus, and Streptococcus showed quite a bit of variations among the kits tested and also between the 16S versus shotgun methods. So these 2 experiments really show the value of whole cell standards, where you can use these whole cell standards to monitor DNA extraction, which could impact your further outcome out of 16S or shotgun analysis. Next, I'm going to show you some of the data we generated based on the genomic DNA mark communities. In this case, we used 2 genomic DNA mark communities, MSA 1000 and MSA 1001, which are a mixture of 10 genomic DNAs. The left side is MSA 1000 data, where we used both 16S and shotgun sequencing for analysis. And the right is for MSA 1001, which is a 10-strain mixture, but in staggered mixture. We got fairly good data with MSA 1000, and we got very good correlation of 16S and shotgun when we mixed all the genomic DNA in equal amounts. However, for MSA 1001, we saw quite a bit of variation in terms of expected versus observed number of % reads. This was quite evident for two bacteria, especially for Rhodobacter and Staphylococcus epidermidis. So this really shows how you can use genomic DNA mark communities to compare various methods such as 16S and shotgun. The next similar experiment we did was for two mark communities, MSA 1002 and MSA 1003, which are more complex genomic DNA mixtures. And we compared even genomic DNA and staggered mixtures side by side with both 16S and shotgun methods. Again, we got a very good correlation between expected versus observed for MSA 1002 and MSA 1003 in some cases, except for Rhodobacter and Staphylococcus epidermidis in this case again. This really shows how you can use genomic DNA to eliminate any bias associated with your method, and how can you use genomic DNA mark communities to standardize any of the steps post-DNA extraction. In the next section, I'm going to show you a couple of examples where we use a standard data set, but we analyzed it on different platforms for analysis. Here we used 16S data set from MSA 1000, which was from genomes in equal proportion, and we tested the analysis against three different database and analysis platforms. We used popular analysis engine QIIME, and against one codex and against NCBI RefSeq. To our surprise, with QIIME and RefSeq database, we could detect only 9 out of 10 bacteria that were in the mixture. We could not detect E. coli at all in the first attempt. And 2 other bacteria, Staph, Epidermidis, and Enterococcus, were barely detectable. RefSeq database could barely identify Lactobacillus gasseri as well. Next, I'm going to show you one example of shotgun data. This was from MSA 1002, which is a mixture of 20 genomic DNA. And in this case, we tested it against 3 different platforms. There was one commercial platform which we tested against, again, NCBI RefSeq database and one codex database. Again, in this, to our surprise, we could not detect 18 out of 20 bacteria with one of the commercial database that we used. RefSeq database could successfully identify all 20 bacteria. However, relative abundance were way off compared to what we were expecting. On the other hand, one codex analysis gave us fairly good ratios of what we were expecting. So again, the purpose of this example is to show how can you use the data sets from standards to even compare various analysis platforms. To summarize, we have developed a set of general microbiome standards in the form of whole cells and genomic DNAs. Whole cells can be used as full process controls to monitor the full microbiome workflow. Whole cells are presented as 10 and 20 organisms mixed with even composition. And the genomic DNA-based standards are very valuable for assay development and for optimization post-DNA extraction. They contain, again, a mixture of 10 and 20 organisms. In addition, they have been mixed as even and staggered composition. One thing which I would like to mention is seeing the successful data analysis with these standards from one codex analysis. We partnered with one codex and we came up with a solution for data analysis as well. All these physical standards come with a standardized data analysis from one codex, which Nick Greenfield is going to talk about in the next section of this webinar. So I'll hand over to Nick.
Great, thanks, Dave. So in this section, I'm going to present a brief overview of what one codex is, and then we'll dive into describing the collaboration with ATCC around the microbiome standards and the analysis that we've built that's tailored to the evaluating these microbiome standards and comes packaged with the standards themselves. And then I'll actually present a demo and show some screenshots of what that analysis looks like and hopefully make it very tangible for everyone. So diving in, a little bit of quick high-level background. OneCodex, we're a software company, as Kara mentioned in the introduction, out in San Francisco, California, primarily a team of software engineers with microbiology expertise, and we've built a platform that's a sequence-to-answer data platform. It's web-based. There's a screenshot here that's really easy to get started with and approachable for anyone ranging from a bench scientist with fairly little computational background to a bioinformatician who may want to do more sophisticated analysis and extend and build on top of the platform. And our platform supports a variety of different analyses, including taxonomic analysis, shotgun classification of metagenomic data, classification of 16S and other common marker gene amplicon data, as well as functional analysis via panels looking for gene content and the like. At a high level, our technology consists of 3 layers. The foundation is a scalable data platform that scales to tens of thousands of samples and lets you build what we think of as data banks of next-gen sequencing samples and allow you to not just use samples in a single experiment, but also return to them, revisit them, do meta-study analyses, and the like. On top of that scalable platform, we have a very strong focus on security and reproducibility and compliance and making sure that every analysis is versioned, every analysis can be reproduced, all changes to data in the platform are logged and auditable, and we see that as being both useful in a research environment, but critically necessary as you start to think about moving from research applications to applications in the clinical space or in drug development. And then on top of that sits a flexible and easy-to-use user interface. We saw a screenshot of that on the prior slide, and our focus, again, is making sure that there's a user interface that's easy and approachable for someone with less computational background in bioinformatics training, and at the same time, they're powerful tools that a bioinformatician or a software developer even can use to extend and build on top of. The platform, our focus is on taxonomic classification as well as functional characterization. So what are the bugs in my sample as well as what are the genes in my sample? And this is all built on top of the largest searchable database of microbial genomes out there. We've got a collection of over 100,000 whole genomes of bacteria, viruses, fungi, protists, archaea, et cetera, that are searchable in the platform and that we can do classification against. Dave highlighted this a little bit earlier in his discussion, but in the course of a sample microbiome study, where we come in is primarily kind of right of the sequencer, if you will, helping with primary as well as secondary analysis into data sets. We also do work with folks and have the ability to facilitate sequencing. And then one final piece of background, and then we'll dive into the microbiome standard, is that here's some recent results from a comprehensive benchmarking survey that came out of Weill Cornell, and this is looking at a couple different accuracy metrics, F1, precision recall, area under the precision recall curve, and this is a very nice study because it's a large comprehensive benchmark of about 30-plus data sets, and across a variety of accuracy metrics. This kind of highlights how well the tool performs relative to some of the other methods that are available out in the literature. So turning now to the ATCC microbiome standards, I think Dave did a nice job setting up the need, but I think a continual and an underlying challenge in microbiome research is inter-laboratory comparability, as well as even sometimes inter-run or inter-lab tech comparability, and really getting a better sense and appreciation for what are the biases in our data. Can we measure them? If we can measure them, can we then hopefully correct for them? So we've worked with ATCC on an innovative kit plus informatics collaboration, if you will, where when you purchase the microbiome standards from ATCC, you'll also get bundled analysis on the OneCodex platform, and that analysis is a scorecard analysis that you see in the top right here that really helps you take this data from a known community, from a control, and say, how well am I doing? Am I sequencing, and am I finding all of the organisms that I expected from a true positive point of view? Am I characterizing their abundance accurately? Am I finding other things that I didn't expect? Do I have contaminants coming into my workflow somewhere? And make that really easy to do, but also very standardized so that you can compare across laboratory protocols, across different lab groups and users, et cetera, and have kind of standardized way of saying, okay, I'm doing well with this protocol at characterizing a mixed community of staggered abundance organisms, 20 staggered abundance organisms, or I'm not doing so well. Let's understand that bias, potentially improve it, decrease it, and certainly be able to measure it. So at a high level, the workflow looks like this, and there are more details on our website as well as on ATCC's website. But we start from the FASTQ file off of the sequencing instrument, and then for shotgun data, we use our OneCodex database. Of whole genomes for 16S data, we use what we call our targeted loci database that has both 16S and other marker genes in it. And we characterize the abundance of the known organisms in the mixture, the MSA 1000, 1001, et cetera products, both for the gDNA or the whole cell products. We characterize the known organism abundances. We use those to derive a true positive score, a relative abundance score, and a false positive score. And I'll show examples of that in a moment as we go through the live demo, but the goal is to kind of capture key components of, did I find what I was looking for? Did I characterize its abundance accurately? And what else comes through? And that's where we're using our OneCodex database and the targeted loci database to really make sure that we're pulling forward reagents, contaminants, anything that may be happening upstream that ends up in your FASTQ file off the sequencer that you didn't expect to be there. Great. And so now I'll turn to a little bit of a demo showing what the ATCC microbiome standard analysis with OneCodex looks like, how to access it, and how to get started. So the first thing I should say is that you'll receive an email if you purchase the ATCC microbiome standards that details some of these instructions and provide you with links to our site and how to log in or register as appropriate. But this is, if you come to our website, it's a web-based application. You can sign up, register an account, just enter your email and a password, and you'll log in and it'll look something like this. You'll be taken first to a page with your samples, and then on the left-hand side, you can see that there's an analysis module that we've built with ATCC for the ATCC microbiome reference standards. And if you click on that, you'll see a page with each of the ATCC microbiome standard products. They're both, you can look at the genomic DNA products, the 1000 series, as well as the whole cell products, the 2000 series. And what you'll do is select a whole cell or genomic DNA product, select which type of sequencing you've done, and then upload the sample and run an analysis. Now, here we see a note at the top that's saying that we need to enter a coupon code. This is what we email to folks when they purchase the microbiome standard. First, the way that we do that is we click the link at the top, redeem the coupon, or over on the left, redeem coupon. And it's a very simple code that's in the email, 6 digits. You enter the code, click redeem, it gets applied to your account. And then, we have some number of uses. We here are going to our desktop, finding the file from the instrument that we want to take a look at, dragging it and uploading it to the OneCodex platform. As simple as kind of dragging and dropping the FASTQ file. Or if you've already uploaded the file, you can select it above from something that has already been uploaded to our platform. The file uploads. For a smaller file, that should be fairly quick on a fast connection. And then after the file has been uploaded, we see it appear up above. There's MSA 1000.16s.example up above. And we have the option to continue and add metadata. So The user is then prompted to answer a very brief survey with some background on how was the sample prepared, if it's 16S, what amplicon region was used, what was the instrument that was used, a couple other questions. It's very short, 5 questions. There's an option for a more detailed survey if you want to collect and capture more metadata about your protocol. But the basic survey is only 5 questions. And you submit that and then click the button for the results. And depending on the size of your sample, you may have to wait a few minutes and there'll be an indicator to tell you so, or the analysis may be done already. And this is what a standard kind of scorecard looks like. In this case, this is for the MSA 1000 product and for 16S sequencing that was done on a MiSeq. And here we see we get an overall score of 89%. That's the average of these 3 subscores. And we get a perfect true positive and false positive score and a not great relative abundance score. And I'll walk through each of those in turn. So the true positive score is quite simply the number of organisms you detected over the number of expected organisms in the input control mixture. The relative abundance score is the correlation coefficient between the number of reads from your sample data and the expected number of reads and content from the control mixture. Typically for 16S data, you're going to see a lower relative abundance score because 16S generates, there's more variability both in PCR and in copy number effects and the like for 16S than there is for shotgun data. So we typically see lower scores, relative abundance scores for 16S than for shotgun data. And there are details on this in the table. And all of these tables can be downloaded, I should say, and filtered and searched and sorted so that they're very easy to explore. And then false positives is the last category. Here we have some kind of trace false positives. Those don't count against the score. But if there are any major false positives where a significant number of reads in the sample mapped to organisms not known to be in the control, that would be flagged and points would be taken out of this score and deducted. And common contaminants you might see there might be like bradyrhizobium, as that can be a common contaminant upstream of sequencing. So that's the overall scorecard. That's how the analysis works and what it looks like. It's a quite simple process of redeeming a code that's emailed with a link when the ATCC microbiome standard ships, logging into an existing OneCodex account or registering a new one, dragging and dropping a file over, or there are options for uploading the data from a command line tool if that's something that you're familiar with and is more helpful, and entering a little bit of metadata and then getting a nice standardized scorecard that says, then answers the question, how am I doing on my microbiome sequencing? And over time, our hope is that also, as you compare different protocols, you can see for these microbiome standards, well, that protocol worked a little bit better. I'm getting better relative abundance estimates and better data in terms of relative abundance when I switch from extraction kit A to extraction kit B, or when I change my library prep protocol, when I use these amplicons for my 16S sequencing, et cetera. So the goal is to provide the data that lets you interrogate and improve those parts of your protocol, and then also, of course, understand the bias in the sequencing data so that you can take it into account, so that you can correct for it, so you can understand both the potential and the limits in looking at your samples of interest. And one last point is on our website, there's a little kind of smiling chat button in the bottom right, here circled in orange, and that's a way, if you ever have any questions about the standard, about the analysis, about your results, if you click that, it'll open up a little chat box that will go to me and the team here, and we're always happy to answer questions, and help you out with your data analysis. So to conclude, we've built a tailored analysis for the ATCC microbiome standards with the goal of helping folks get fast, easy to use results, but also really good, high-quality analysis that's tailored to the question of, am I finding and detecting my control mixture accurately? and lets you validate and optimize your wet lab protocols, as well as better understand both the potential and limits of your data on the dry side. So with that, I'll hand it back to Dave.
Thank you, Nick. You can find more information about ATCC microbiome standards by visiting atcc.org/microbiome. In addition to the whole cells and genomic DNA-based standards, we do have individual strains and nucleic acid standards as well. And of course, we provide bioinformatic tools in the form of one codex for analysis of these microbiome standards. Thank you very much.
Thank you, Dr. Mittar and Mr. Greenfield, for the excellent presentation, and thank you everyone for attending the webinar. Please visit us online at www.atcc.org/webinars to register for additional ATCC webinars. For more information about the microbiome standards or about the One Codex platform that were discussed in today's webinar, please visit us online at www.atcc.org/microbiome and www.atcc.org/bioinformatics. Also, we would love to hear your feedback. Please take our survey after the webinar ends to let us know what type of microbiome standards you need for your research. Thank you again, and have a great day.